My study wished to address the impact of different types of leaf litter from native deciduous trees in southern New England on D. hortensis populations. First of all, it's important to define what exactly D. hortensis and deciduous trees are. D. hortensis is a species of earthworm, colloquially known as the European nightcrawler. They are commonly used for fishing and composting and can be currently found within New England soils. Deciduous trees are trees which shed their leaves in preparation for winter. As the leaves accumulate on the ground, they form what is called a leaf litter. Earthworms, the majority of earthworms in New England are not native to the area, having been wiped out due to glacial spread 10,000 years ago during the Ice Age. The majority of earthworms, such as D. hortensis, have come from Europe and Asia through settlements and trade, and have dispersed throughout New England forests through agriculture, logging, and fishing. Earthworms are decomposers, which means that they eat dead and decaying matter. There are three main types of earthworms which eat dead and decaying matter from different parts of the soil. Epigenic earthworms eat organic matter from the surface of the soil. Endogenic earthworms eat organic matter within the soil, and anesthetic earthworms eat both organic matter from the top of the soil and within the soil. European nightcrawlers are anesthetic earthworms, which I chose because of their relationship with the leaf litter. There are many different types of native trees to New England. The two main tree genre are oak/hickory and maple/beech/birch trees. Specifically from those, I chose the red maple, sugar maple, and northern red oak, because the more prevalent a tree is within populations and within the forest, the more common their leaf litter will be. So I found that those would be the most applicable and relevant. So it is known that the increases in nutritional availability for both plants and animals results in increases in growth and reproduction. This is because with no limitations, and at least less limitations to one's growth and ability to grow and reproduce, animals are better able to reach their biotic potential. And how this relates to both earthworms and leaf litter. Leaf litter is, as I said earlier, a source of food for earthworms, typically falling in between a carbon to nitrogen range from 20:1 to 30:1. This is sufficiently nutritionally sufficient for earthworms, as they fall into a range from 25:1 to 30:1. However, the study by Januszewski and Striti found that carbon to nitrogen ratios as leaf litter was decomposing actually varies based on leaf type. This raises questions as to how different leaf types may have different influences on the earthworms based on those variations in those carbon to nitrogen ratios. So first of all, we have to examine why this discussion actually matters. Earthworms have had a large impact on the environment. As I said earlier, the majority of earthworms are not native to the area. Before their invasion into New England soils were categorized by slow rates of nutrient cycling, as it had greater soil stratification and a well-developed organic horizon. Essentially, the layers of the soil were more distinct from one another. However, after earthworms invaded, earthworms have impacted the soil by resulting in changes to that soil structure. They have increased calcium, magnesium, potassium, and phosphorus cycling rates and have reduced fungal dominance. This has led to alterations in soil biogeochemistry, decreases in below-ground biodiversity, essentially the biodiversity of the soil, increases in understory populations, as the soil is now more habitable for understory populations, decreases in desirable floral populations, and actually making the soil more habitable for invasive species. While there is a lot of information on the impact of earthworms on the environment, there is much less on the impact of the environment, variations in the environment, on earthworms. The only existing study I could find, one of the only studies I could find regarding this, is a study performed by Magrath and Lindroth, where they examined the impact of different leaf litter from invasive plant populations on invasive earthworm populations. They found that the invasive plants which they studied, honeysuckle and buckthorn, actually have high quality leaf litter which more stimulate invasive earthworm populations. This leads to a positive feedback loop, as increases in earthworm populations based on higher quality leaf litter increased earthworm populations and increases in earthworm populations make soil more habitable for invasive plants, pushing our New England forest even further away from pre-colonization environment. So overall, this leads to a gap in knowledge. While there's information on the impact of non-native earthworms on the environment, and there's information on the impact of invasive species on flora, on earthworms, there's no information currently on the impact of native plants, variations in native plants on earthworms, which is what I wanted to examine within my study, leading me to my research question. How do different types of leaf litter from native deciduous trees in New England impact these D. hortensis populations? To examine this, I chose a true experimental design. True experimental designs are where you cause variations to a single independent variable. And then you have a control group to compare the impacts upon those different variations have on the structure as a whole. The reason why I chose this design was because it has a very high, strong internal validity, which means that I was able to create to determine a causal relationship between the types of leaf litter and the earthworm populations. So the materials I sourced, I had European night crawlers that I obtained from Brothers Worm Farm, leaf litter I collected from local New England forests, bagged fertilizer-free topsoil, and 4 plastic clear containers. To set up these individual containers, I put 2,200 grams of bagged topsoil and 40 grams of leaf litter per container. And again, the 4 different types of leaf litter I chose were the red maple, sugar maple, red oak, and then the mixed/control group. And the reason why I chose these specific amounts of leaf litter and topsoil was because I needed to get the depth to a sufficient range for D. hortensis, which is between 6 to 12 inches deep. I did 3 measurements over the course of my 8-week study. First at the beginning, 1 in the middle, and 1 at the end. So thus, each section was during a 4-week gap period between each measurement. To maintain the soil and to actually make sure that the worms were in a healthy environment, I watered the soil 2 to 3 times a week to make sure the soil was moist enough for them to survive, as well as to better mimic the New England soils, which have high rates of precipitation and thus moisture soils. There are a few limitations to my methodology, primarily time and resources. Time because it's a study addressing growth and reproduction, which is typically done on a yearly scale. And as I was only limited to the 8 weeks that I was given, I have less information and less data points on the actual projections of growth and reproduction for the worms. Secondly, resources. Due to financial and time constraints, again, I was not able to obtain the resources needed to create the perfect methodology. In an ideal world, we would have had more soil and more worms to examine and more different types of trees. But as I had to purchase these leaves and I had to obtain these leaves as well as all the other resources, it led to limitations that I had to adjust for. So overall, even though Janisowski and Striti found that there were carbon to nitrogen ratios variations within leaf litter, I figured that these variations would all fall under ideal range for earthworms and would not lead to any significant impact upon their growth and reproduction, leading me to the hypothesis that there would be no significant impact of leaf litter type on earthworm growth and reproduction. However, this was actually contrasted by my results. My results found that for earthworm mass, that red maple had an initial decline in mass while having an incline later on. Mixed control group initially peaked before decreasing, while sugar maple and red oak stayed fairly consistent throughout the study, hovering around 90 grams of mass. I also found cocoon number varied greatly between each section, between each container. The red maple group was the outlier, having 20 cocoons at the conclusion of the study, While the sugar maple only had 3, the red maple and mixed control group had 8. These variations in this earthworm mass, which was used to determine what the actual growth of the earthworms were, because the more you grow, the greater your mass is, and the variations in earthworm cocoon number, which I used to indicate reproduction, lead to the conclusion that yes, leaf litter does actually have an impact on earthworm growth and reproduction. However, what kind of impact? It is inconclusive. Again, I had limitations to my data, primarily due to time, and I was unable to determine any consistent trend or statistical significance from my results. There are a few implications of my results. As I said earlier, earthworms have a large impact on the environment, and they might be inadvertently impacted through logging and forest management. Logging and forest management, they tend to either prioritize certain trees due to biodiversity or tree profit considerations. So loggers or people in charge of forest management may prioritize certain trees or change current rates of biodiversity, which may actually have an impact on earthworms and how much they're able to reproduce and spread, because the more of a certain type of tree, if that tree is more nutritionally beneficial, you may be inadvertently increasing the spread of earthworms in their growth and reproduction. There are some limitations to my conclusion and my results, however. Even though this does have large implications for the logging and for the forest management, it's pretty restricted due to the fact, I chose to focus specifically on leaf litter. As I said earlier, earthworms are decomposers, and they'll eat dead and decaying matter from all different parts of the tree, such as their roots, their branches, their bark. I did not take into consideration those different parts of a tree. I made that choice within my methodology because to limit the scope of my study, and I did not find any existing literature on variations between bark nutritional values. Secondly, my region studied. I chose New England, which is hyper-specific in the fact that the majority of earthworms are non-native. Other parts of the United States, which are not as glaciated as New England, they tend to have more native earthworms. So they might have different relationships or being impacted very differently by the presence of these non-native earthworms compared to New England, because those areas are likely more adjusted, the forests are more adjusted to earthworm presence. And C, earthworm type. Again, I chose European earthworm, which is an antecedent earthworm. I did not address or study epigenetic or endogenic earthworms. This may not, since those species of earthworms have different relationships with leaf litter and organic matter, then these results may be completely different for those species types. So overall, why does this matter? Determining how different factors influence these earthworm populations can help us mitigate earthworm spread by showing what trees we should prioritize and which trees we should not prioritize due to their impact on earthworm populations. And secondly, it can help us better develop projections of earthworm distribution and spread. Because if there's certain forests or areas that have a higher percentage of a tree type that is more nutritionally beneficial for earthworms, we can predict that they will have higher rates of earthworm spread. Lastly, this leads to my conclusion that actually different types of leaf litter from deciduous trees that are native to New England do impact the growth and reproduction of D. hortensis differently. However, due to the limitations of my study, it does lead to a few questions that can be addressed in the future, such as A, what specific nutritional differences in leaf litter cause these earthworm population variations? I assumed it was the carbon to nitrogen ratios because of those variations, but it may have to do with another component of the desiccated leaf litter that I did not take into consideration, as well as what are the impacts of leaf litter from other organic sources, such as undergrowth and coniferous trees, on earthworm growth and reproduction, as they also constitute a portion of the leaf litter that you can find on the ground. Overall, I hope these questions will be explored in the future and give better insight as to how to prevent and best manage earthworm spread and reproduction. Thank you.
Thank you. How did your initial exploration of the scholarly conversation lead to your final research question?
Okay, so my initial examination of this study is I did not know that there were any variations within leaf litter type. And going into the scholarly discussion, I was under the assumption that earthworms were actually beneficial for the environment. So my integration into the scholarly information and existing literature out there gave me insight as to how earthworms are actually detrimental to the forest, which shaped my research question into pointing into ways that you could possibly, my project goal into ways that you can possibly predict their spread or thus mitigate their spread going into the future.
How do your findings provide direction for future research in the field?
Well, my findings actually indicate that there are variations due to leaf litter type. This leads to many different ways that you can go, routes you can go, such as examining not how these variations can actually impact other animal species or other decomposers within the soil or any other animal or species that eats this leaf litter, how they are also impacted by this, these variations. As again, I also said here, it can also go into information as to how like variations in leaf litter, like what specifically those nutritional values. So it gives insight into maybe further examining these differences within leaf litter.
Thank you.
Think back to the initial curiosity that sparked your inquiry. What other curiosities do you have, and how has this process prepared you to explore them?
Wonderful. I have a lot of curiosities going out of this. My initial curiosity was, how do earthworms actually interact with the environment? How are they impacted by the environment, or how do they impact the environment? And I found that in my study that they have a negative impact on the environment and that they are also impacted by the environment. However, I still have a lot of questions as to curiosities as to, like I said earlier, what exactly these differences are, like that make these variations so great, as well as addressing like how other animal species are influenced by it, because I am personally interested in the field of environmental science, and I am concerned as to how these variations may impact other important native or invasive species. This process has allowed me to know and learn how to develop a methodology and to how to best explain and develop my ideas, and to create studies that may be similar to this one if I ever wish to explore worms in the future. And so I hope that through these methods and the ways that I learned through this project, I'm able to continue to explore these curiosities in the future.
Thanks.